Download this code from codegib.com, link in the description below. OK, let's delve into a comprehensive tutorial on running the Ncbi prokaryotic genome annotation pipeline, PGAP, on your own data. This will be a detailed guide, covering everything from data preparation to interpreting the results. I'll provide code examples where applicable, especially for tasks like sequence manipulation and data conversion. Keep in mind that running PGAP often requires substantial computational resources. Introduction to Ncbi PGAP. The Ncbi prokaryotic genome annotation pipeline, PGAP, is a powerful, automated system designed to annotate prokaryotic genomes, bacteria and archaea. It performs a variety of tasks, including genome assembly quality assessment, assessing the quality and completeness of the assembled genome, coding sequence, CDs, prediction, identifying potential protein coding genes within the genome. This is a crucial step. Functional annotation, assigning functions to the predicted proteins based on sequence similarity searches and other bioinformatic methods. RNA gene prediction, identifying ribosomal RNAs, rRNAs, transfer RNAs, tRNAs, and other non-coding RNAs. Pseudogene identification, identifying genes that appear to be non-functional due to mutations or other disruptions. Quality control and reporting, generating reports summarizing the annotation results and providing information on the data quality. Why use PGAP? Standardized annotation, PGAP provides a consistent and well-documented annotation. Automation, it automates a complex process, saving time and reducing manual effort. Integration with Ncbi resources, PGAP seamlessly integrates with other Ncbi databases, allowing you to easily access related information. High throughput, designed to handle large volumes of genomic data. 2. Prerequisites. Before you start, ensure you have the following. 1. A prokaryotic genome sequence. This is the core requirement. It should ideally be in FASTA format. 2. Appropriate computing resources. PGAP is computationally intensive. You'll need a machine with sufficient memory, at least 8 to 16 GB recommended, and processing power. Consider using a server or a cloud-based environment. 3. Ncbi dataset CLI tools. You will need to install these tools so you can download the PGAP executable, and its dependencies. 4. Command line proficiency. You need to be comfortable with using the command line in a Linux or Mac OS environment. 5. Optional, basic bioinformatics knowledge. Understanding basic concepts in genomics, sequence alignment, and protein function will be helpful for interpreting the results. 6. PGAP executables and dependencies. You will need to download these using the Ncbi datasets tool command. 7. Install the necessary data packages using the PGAP executable. 3. Data preparation. This is a critical step. The quality of your input data directly affects the quality of the annotation. 1. Sequence format. FASTA format is essential. The genome sequence must be in FASTA format. A FASTA file is a plain text file with sequence entries, each starting with a character followed by a sequence identifier, example sequence 1. The sequence itself follows on subsequent lines. 2. Sequence cleaning. Remove ambiguity codes. Ideally, your sequence should only contain A, T, G, and C characters. If you have ambiguity codes, example N for unknown, R for A or G, consider replacing them with N. A few ambiguity codes are usually tolerated, but too many can affect the accuracy of the annotation. Asterisk remove gaps. Asterisk if your sequence contains large gaps represented by hyphens, or other characters, consider removing them or replacing them with n. Asterisk check for vector contamination. Asterisk if your sequence was generated by cloning, check for vector contamination and remove it. Tools like VecScreen, available at Ncbi, can help. 3. Assembly quality. Asterisk contig count. Asterisk a finished genome should be a single contig, chromosome. If you have multiple contigs, that's okay, but PGAP will treat each contig as a separate chromosome or plasmid. A highly fragmented assembly, many small contigs, can make annotation more difficult and potentially less accurate. Asterisk contig length. Asterisk very short contigs, example less than 500 BP, are generally not suitable for annotation. 4. Naming conventions. Asterisk contig names. Asterisk ensure the sequence identifiers, after the character in the FASTA file, are meaningful and unique. Avoid special characters in the names. Try to keep them relatively short and descriptive. 5. Data conversion, if needed. Asterisk if your sequence is in a different format, example GenBank, you can convert it to FASTA using tools like Secret from the Emboss suite. Asterisk you can also use BioPython, a Python library, for sequence format conversion. 4. Running PGAP locally, command line. This is the central part of the tutorial. Here's how to run PGAP using the command line, step by step. 1. Install Ncbi dataset CLI tools. Asterisk. Follow the instructions on the Ncbi website for installing the Ncbi datasets command line tools. The exact steps will depend on your operating system. A typical installation on Linux or Mac OS might involve downloading a binary and adding it to your path. Follow the instruction on the Ncbi website to install the datasets tool. Asterisk Ncbi datasets documentation, https://www.ncbi.nlm.nih.gov/data/zts/docs/v2/download-install. 2. Download PGAP executables and dependencies. Asterisk. Use the datasets command to download the PGAP package. This will download the PGAP executable and all its required dependencies. First, create an installation directory. 3. Install the data packages required by the PGAP executable. Run the PGAP installation routine. 4. Set up environment variables, important. PGAP requires several environment variables to be set correctly. These variables tell PGAP where to find its data files and other dependencies. You can set these variables in your .bashrc or .zshrc file, or directly in your terminal session. Replace /path/to/your/pgap_install_directory with the actual path to the directory where you downloaded and extracted the PGAP package. The Ncbi_dataset folder should be located inside the pgap_install folder from the last step. These will be something like /home/username/pgap_install/ncbi_dataset. Explanation of environment variables. **PGAP_data, specifies the location of the PGAP data files, example protein databases, scoring matrices. **PGAP_config_dir, specifies the location of PGAP configuration files, example parameters for various steps in the pipeline. *BLAST, specifies the location of the BLAST databases used for sequence similarity searches. PGAP relies heavily on BLAST. Important **. After setting these environment variables in your .bashrc or .zshrc file, you need to source the file for the changes to take effect. 5. Run PGAP asterisk. Now you can run PGAP on your genome sequence. The basic command is explanation of command line options asterisk. Asterisk in specifies the input FASTA file containing your genome sequence. Asterisk out specifies the output directory where PGAP will store its results. PGAP will create this directory if it doesn't exist. Asterisk organism specifies the name of your organism. This is very important. PGAP uses this information to select the appropriate genetic code, taxonomic information, and other parameters. Use the scientific name, example Escherichia coli. Other useful options asterisk. Asterisk prefix specifies a prefix to be added to the names of the output files. Asterisk CPUs specifies the number of CPU cores to use. This can significantly speed up the annotation process, especially on multi-core machines. For example, CPUs 8 would use 8 cores. Asterisk config select allows you to specify a subset of contigs to annotate, example config select config 1 config 2. Asterisk taxid specifies the Ncbi taxonomy ID for the organism. This is an alternative to organism. You can find the taxonomy ID on the Ncbi taxonomy browser, https://www.ncbi.nlm.nih.gov/taxonomy. Asterisk domain specifies the domain of life, bacteria or archaea. PGAP can usually infer this from the organism name, but you can specify it explicitly if needed. Asterisk euc_model allows you to set this to false to ensure that PGAP does not attempt to use eukaryotic annotation models on the sequence. Example with multiple cores asterisk. 6. Monitor the progress asterisk. PGAP will print messages to the terminal as it runs. It can take a significant amount of time to complete, depending on the size of the genome and the available resources. Be patient. You can use the top or stop command to monitor CPU and memory usage. 7. Troubleshooting asterisk. Asterisk check the error messages asterisk. If PGAP encounters an error, it will print an error message to the terminal. Read the error message carefully to understand the problem. Asterisk check the log files asterisk. PGAP generates log files in the output directory. These files can contain more detailed information about the errors. Asterisk environment variables asterisk. Double check that your environment variables are set correctly. This is a common source of problems. Asterisk permissions asterisk. Make sure you have right permissions to the output directory. Asterisk dependencies asterisk. Ensure that all the necessary dependencies are installed. Asterisk contact Ncbi support asterisk. If you're still having problems, you can contact Ncbi support for assistance. PGAP output and interpretation. Once PGAP completes, it will generate a directory, specified by the out option, containing the annotation results. The most important files include *.gff, general feature format. This file contains the annotation information in GFF format. It describes the location of genes, RNAs, and other features on the genome. This is the primary file for downstream analysis. *.fna, FASTA nucleotide contains the genomic sequence. *.faa, FASTA amino acid contains the amino acid sequences of the predicted proteins. *.fnt, FASTA nucleotide translated contains the nucleotide sequences of the predicted proteins. *.tbl, feature table. This file contains a table of features suitable for submission to GenBank. *assembly.report.txt contains information about the assembly, including the number of contigs, total length, and N50. Asterisk statistics.txt asterisk provides statistics about the annotation, such as the number of predicted genes, the average gene length, and the GC content. Asterisk errors.txt asterisk contains error messages encountered during the annotation process. Asterisk warnings.txt asterisk contains warning messages encountered during the annotation process. Interpreting the results asterisk. 1. GFF file analysis asterisk. The GFF file is the most important output file. You can use bioinformatic tools like Artemis, Ncbi, Integrative Genomics Viewer, or custom scripts to visualize and analyze the annotation. The GFF file contains information about the coordinates of each feature, its type, example gene, CDs, tRNA, and its attributes, example gene name, product name. 2. Functional annotation asterisk. Look at the product names assigned to the predicted proteins in the GFF file. This will give you an idea of the functions of the genes in your genome. PGAP uses sequence similarity searches, BLAST, against protein databases to assign functions. 3. RNA genes asterisk. Check for the presence of rRNA and tRNA genes. These are essential for protein synthesis. 4. Pseudogenes asterisk. Examine the pseudogenes that PGAP has identified. Pseudogenes are genes that have lost their function due to mutations or other disruptions. 5. Assembly quality asterisk. Review the assembly.report.txt file to assess the quality of the assembly. A higher N50 value and a lower number of contigs generally indicate a better assembly. 6. Statistics asterisk. Use the statistics.txt file to get an overview of the annotation results. The number of predicted genes and the GC content can provide insights into the genome. Example, analyzing the GFF file with Python. This Python script will 1. Read the GFF file. 2. Parse each line, extracting the relevant information. 3. Create a pandas data frame to store the data. 4. Print the first few rows of the data frame. You can then use pandas to perform various analyses on the GFF data, such as filtering features, calculating lengths, and counting the number of genes. 6. Advanced topics. Asterisk custom databases asterisk. PGAP allows you to use custom protein databases for annotation. This can be useful if you have a specialized database of proteins related to your organism. You need to format the database correctly for BLAST and configure PGAP to use it. Asterisk manual curation asterisk. While PGAP is a powerful tool, it's not perfect. Manual curation of the annotation is often necessary to correct errors and improve the accuracy of the results. You can use tools like Artemis to manually edit the GFF file. Asterisk genome submission asterisk. If you plan to submit your genome to GenBank, you'll need to prepare the data in the correct format and provide the necessary metadata. PGAP can help with this process, but you'll need to review the data carefully and make any necessary corrections. 7. Conclusion. Running PGAP locally can be a complex process, but it gives you full control over the annotation pipeline and allows you to customize it to your specific needs. By following the steps outlined in this tutorial, you should be able to annotate your own prokaryotic genomes using PGAP and gain valuable insights into their biology. Remember to consult the Ncbi documentation and seek help from the community if you encounter any problems. Good luck.